Okay. So adaptive defenses in the house. Okay, so now we're past the first two lines of defenses. Okay. Now we're into adaptive immunity. Adaptive immunity are defenses that target a specific pathogen. One of the characteristics of the first two lines of defense is that it doesn't really care about what type of pathogen you really are. It treats viruses, bacteria, everything the same way. Now, you care very, very much about what type of pathogen you are. Okay. And so it requires infection or vaccination. And there is a big difference between primary response and secondary response. And I'll explain more about that in a little bit in the chapter. Okay.
Host defenses consist of two categories, the innate or non-specific.
So sharing.
Defenses and the adaptive or specific defenses. The human body has many innate defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, phagocytosis of microorganisms, inflammation, and the complement system. The human body has two main divisions of adaptive defense, humoral immunity and cell-mediated immunity. Antigen-presenting cells aid in the activation of these defenses. A humoral immune response consists of the activity of B cells and the antibodies they produce. Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups, helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The innate and adaptive components of the immune system work together to carry out host defenses.
Okay. So, this objective is definitely important. Okay, so from the point of view of your immune system, there's two things about you. Okay, the cells that make up who you are and the fluid that surrounds those cells. Okay, so humoral immunity and cellular immunity. Humoral is fluid. Okay, the ancient word for fluid is humor. Okay, so humoral immunity is regulated by B cells. Okay, and B cells produce antibodies that combat molecules known as antigens. Antigen isn't necessarily bad. Okay, antigen is anything that generates an antibody. Antibody, so antigen is antibody generating. Okay. Okay, so B cells are lymphocytes that are created and mature in the bone marrow. Okay, they recognize specific antigens and they make antibodies to those antigens. Okay, they're named for the bursa of Hemorrhagus in birds where they were first discovered, but that's not really that relevant. Okay.
The adaptive immune response is divided into two categories, humoral immunity and cell-mediated immunity. A humoral immune response consists of the activity of B cells and the antibodies they produce.
Okay, the other side is the cells. Cells are controlled by cellular immunity is controlled by T cells or T lymphocytes. Okay, these guys mature in the thymus instead of the bone marrow. So B for bone marrow, T for thymus. Okay, T cells have T cell receptors on the surface that contact antigens, causing T cells to secrete cytokines instead of antibodies. So T cells will literally come up to you and kill you instead of secreting antibodies. they come up to the cell and they secrete cytokines that will kill the cell. Okay. Okay. So where these guys actually work is not really the bloodstream, but lymphoid tissue like bone marrow. Not bone marrow. Sorry, spleen, lymph nodes, etc., etc. Okay, hypophages, tonsils. Okay, but mostly the lymph nodes. That's why they're called lymphocytes, because they're mostly in the lymph nodes. Okay. Okay. So humoral immunity, fluid, cellular immunity, cells. Okay. Okay. Make sure you understand this question. Okay. Okay. A cytokine, by definition, is a chemical messenger produced in response to a stimulus. Okay. Interleukins, okay, are cytokines that work between leukocytes. So they usually have IL and then a number, IL-1, IL-2, IL-14, etc., etc. Okay. Chemokine will induce migration of leukocytes. Okay. So they know where to go. They find the signal by going up the concentration of chemokines. Okay. Interferons and TNF alpha we already covered in the last chapter. Okay, so I won't go over those again. Okay. Hematopoietic cytokines control stem cells that develop into white blood cells and red blood cells. Okay. Overproduction of cytokines lead to a cytokine storm, which throws your whole entire body out of whack and can actually kill you. Okay. I have some fear that there will be people who actually will get COVID-19 again, and they might get a cytokine storm again and die from it. The disease that we most compare COVID-19 to is this flu of 1918, right? That was the last time there was this big pandemic. And what happened was a lot of healthy young people in their 20s, like you guys, actually died from exposure to flu virus because they generated a cytokine storm. The response was so big and generated so much of a response in their bodies that actually killed them. Okay, so that's something to actually think about and look out for. Okay. Honestly, I haven't heard anything new about cytokine storms in COVID-19 patients for 6 months now. But in the beginning, that was something that they were really concerned about. But yeah, I should check up on the news on that later. Okay. So here are some key cytokines. Okay. 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 These 3 objectives, all of them, definitely on the test. Okay, without a doubt. So make sure you learn these 3 objectives. Okay. Okay. An antigen. Okay. I don't know what the typo is. It's usually components of invading microbes or foreign substances. Okay. And it could be anything. It could be COVID-19, it could be flu virus, it could be pollen, it could be cat hair, it could be shrimp, anything that generates an antibody response. Okay. Haptenings are used when the antigens themselves are too small to provoke an immune response. And we purposely attach them to haptenings, and then they generate immune responses because the antigen has to be a certain size to actually generate an immune response. Okay. So here is a bacteria. Okay, a bacteria is not an antigen. A bacteria has antigens. Okay, so this protein on the surface of the bacteria might be an antigen. Okay, just because it's an antigen, okay, means it has at least one epitope. An antibody will attach to a particular antibody will attach to one particular epitope. Okay, so like antibody A will attach to the triangles. Antibody B, okay, will attach to these little things right here. Antibody C will attach to the squares, etc., etc. Okay. This is happening. Okay. Okay. Antibodies. Okay. Antibodies are a bunch of proteins called immunoglobulins. Okay. Valence is the number of antibody antigen binding sites. Okay. Most antibodies, the ones that you guys probably already know what antibody is mostly, and the ones you probably picture in your head is the bivalent antibodies with 2 binding sites. Okay, there are some with more. Okay, all right. So this is a typical antibody. Okay, it's an IgG antibody. Okay, about 80% of your antibodies in your blood are IgG. So this is the IgG antibody. Okay, now let's talk about the important parts. Okay, the parts that's first of all, there are 2 chains. There is a heavy chain and there's a light chain. Okay, the green is a heavy chain, the blue is a light chain. That's just based on size. The heavy chain is bigger. Okay, in the heavy chain and in the light chain, there are 2 separate regions. Okay, a constant region that is the same for every single antibody, and an antigen binding region that is different for every single antibody. Okay, this side of the thing, okay, the constant region will respond to macrophages. It will respond to the membrane attack complex, so complement. Okay, it will attract neutrophils because it's all the same. So they're macrophages and neutrophils are trained to recognize the constant region of the antibody. Okay, the antigen binding sites are going to be different for every single B cell. Okay, and that will allow all these B cells, each one to bind a slightly different antigen. So all the B cells in your body will work slightly differently and attack a slightly different antigen. That's what gives you immunity to all these different antigens. Okay, that's why you can fight off a variety of diseases. Okay. Okay, so here you can see the antigen actually attaches to, or the epitope, okay, attaches to the antigen binding region of the antibody. Okay. Okay. So the 5 different types of antibodies. Okay, the mnemonic. Okay. MDs give everybody aspirin. Okay. Okay. So that's the mnemonic. Okay. I'm gonna talk about them in order because they actually make sense in order. There's a reason why this is a mnemonic actually. Okay. So first thing to talk about is IgMs. Okay. Oops. I don't know what the hell is going on here. Okay. IgMs. Okay. IgMs are made out of 5 monomers held together by a J chain. Okay. Let me see if I can find a picture of IgMs. Yeah. Okay. So these are IgMs. Okay. So these are 5 monomers held together by a chain here, and so you have 10 binding sites for antigens, really. Okay. So one of the things that this antibody is very good at is holding things together. So each of these can hold 2 antigens, right? So this whole chain can hold 10 antigens. That's very likely, but it can. But most likely it'll hold like 2 or 3 different bacteria together. And if you attach them to different types of the bacteria, then the whole thing clumps together. Okay, and the whole thing clumps together. If all the bacteria are clumped together and can't move and stuff like that, then the whole thing can be swallowed by one big macrophage. Okay, and it will stop the disease from spreading. It will stop the virus from spreading. It will stop the bacteria from spreading. Okay, so it's especially effective against microorganisms and agglutinating antigen. That's what it means. Okay, it is the first antibody response produced. Okay. Okay, IgG is the most rarest one. It doesn't really have a great defined function. It's actually the one that's stuck on the surface of the B cell. Okay. IgG is the most common one. Okay. It's the secondary. So it's the one that comes out second. Okay. So in a lot of the tests for COVID-19 antibodies, okay, they have 2 separate bags. One bag looks for IgM and one bag looks for IgG. Okay. Okay. So IgG antibody, it crosses the placenta, protects the fetus. Okay. It triggers complement. It has this phagocytosis. It neutralizes toxins and viruses. If it binds to the toxin, it'll prevent the toxin from actually working. Okay. So antitoxin. Okay. Viruses. Remember, viruses have to attach and do undergo this whole process to get in. If it can attach to the binding site of the virus, then that prevents the virus from binding to the cell. Okay. All right. MDGEA. Okay. IgEs, okay, are mast cells, basophils, and blood. They actually cause the release of histamines when bound to antigen. Okay, they also cause the lysis of parasitic worms. Okay, IgA. We talked a little bit about IgA when we talked about some bacterial responses, and one of them that there's an IgA protease. IgA is a dimer in secretion. That's the most important thing. IgA is an antibody that is secreted. Okay, it goes to outside the body. Okay, it goes in your GI tract. It goes in your mucous membranes. It goes in your saliva. Okay, it also comes out in breast milk. So when breastfeeding, you're providing IgA to your child. Okay. Okay. No. Oh. Clear. All right. Okay. Actually. Okay. MHC, major histocompatibility complex. Okay. So MHC genes encode molecules on the cell surface. Okay. Every nucleated animal cell has MHC1 molecules on the surface. Okay. They help identify self. Okay. So a white blood cell will come along and make sure that you have MHC1 molecules on your surface. Okay. What will happen, or a T cell will do that. So I'm going to be more specific. This is where you need to be more specific. So they'll come and identify itself. Okay. What they'll also come and do, okay, is check what it means by self. Okay. So anything that happens to the cell, there are some markers that are displayed on the MHC1. Everything you eat, everything you digest, all the like your mitochondria, everything that ends up in the lysosome will show itself on MHC1. Okay, so if a virus gets inside you, it will show up on the MHC1. Bacteria gets inside you, it will show up on the MHC1. Okay, that's how your immune system will know that you're infected. No matter what goes inside you, it will show up on MHC1. Okay? Okay. So it helps identify stuff. Okay. So these T cells will come along and check not only do you have the same MHC1, that whatever else you're displaying on the MHC1 matches is acceptable, basically. Okay. Okay. MHC type 2 or class 2 are the surface of something called a antigen presenting cell. Okay. There are a few types of antigen presenting cells: B cells, okay, macrophages, dendritic cells. They're all antigen presenting cells. Okay. Okay. Inactive B cells contain a surface IgD that binds to antigen. Okay, the B cell internalizes and processes the antigen that's there. The antigen fragments are displayed on MHC class 2. Okay. Okay. The T helper cell connects the displayed antigen fragment and releases cytokines that activate the B cell. Okay, so here. Okay. So let's look at these B cells right here. These B cells will bind to it if there's an infection. That one of the B cells will bind to the antigen. Okay, just randomly, one of the B cells will bind to the antigen. The B cell, once it binds to the antigen, will intake the antigen, and it will chop up the antigen into little bits. Okay, it'll be like, okay, I bound to something that's foreign. I need to know how to kill this. So I'm gonna take it in. I'm gonna chop it up into little bits, and I'm gonna display all the little bits on my surface. Okay, now all the little bits on the surface of the B cell, T helper cells are come along and goes, okay, I'm checking. So this is MHC class 2. So the T helper cell will check out the MHC class 2 and go, oh shit, okay, now I have a job to do. Okay, usually it will check the B cell, and if there's nothing displayed on there, then it doesn't care. But now that there is an antigen on there, okay, now I do care. Now I'm gonna be like, okay, this B cell has a job to do, and therefore I will send it a signal to make more of itself. Okay, so this B cell will start making thousands of copies of itself. Okay, that process is called clonal expansion. Okay, clonal expansion will make thousands of B cells. About 80% of those will become called plasma cells and start secreting antibodies. So instead of having IgG on the front, it will start making IgG and IgM. Okay, it will make thousands and thousands of antibodies. All of the antibodies that's made binds to the square. Okay, so if this happens, then I have antibodies that attack the squares. Okay, and I will have another B cell down here that bind that the antibody bound to the triangle, or triangle, the circle. Okay, and then I'll have the same response. So now I have plasma cells that attack the squares and plasma cells that attack the circles. Okay, that's how your immune response works. That's how your humoral immune response works. Okay. Okay. Clonal selection differentiates the activated B cells into antibody cells and memory cells. Okay. Clonal deletion eliminates the harmful B cells. Occasionally, you have B cells that bind, and this happens very early in development of B cells. It binds to something that it's not supposed to, and then it commits suicide. Okay, and that's clonal deletion. Okay. 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 So I have a stem cell. This stem cell differentiates into B cells. Every single B cell will have a different variable region. Okay, so it will have it recognize a different epitope. If this epitope doesn't recognize anything, nothing happens. Okay, if it does recognize something, it undergoes it will intake it. It will signal a T helper cell. A T helper cell will give it the signal to start differentiating, and you will make a lot of memory cells and much more plasma cells. Okay, this is about 80 to 20. Okay, the memory cells don't do anything. Okay, the plasma cells will secrete a whole bunch of antibodies. Okay. Okay. Activation and clonal expansion of antibody producing cells. Okay. So T-dependent antigen is an antigen that requires T helper cells to produce antibodies. Okay. T-independent antigens. Similar to B cell without help of T cells. Usually that's because you have this repeating epitope that happens a lot, and it binds to multiple sites of a B cell, and that independently will trigger the B cell to go and proliferate. But here's the thing. There's no T cell response. So the response will be weak, and there's no memory response. Okay? I will try to tie this back to COVID-19. Okay? One of the things that people were worried about were people's antibodies disappearing after getting COVID and testing positive for COVID antibodies. Okay, and all of a sudden the levels drop precipitously. There's some slight evidence that people are able to get COVID again a couple months later. Well, I don't know how well that data holds up yet. I'm still looking at it. Okay, but I truly believe some of them do. Okay, and the reason is these are the T-independent antigens. Okay, that the T-independent antigens are usually polysaccharides. Okay, repeating units of sugars have these little spikes on them that repeatedly bind to epitopes and trigger the T-independent response. Okay, but the T-independent response is really weak, and there's no memory. So you get it, you make antibodies, and then it dies off, and there's no memory of it. So that leaves you vulnerable to a second infection. Okay, that's what we're worried about. Okay, all right.
Certain phagocytic cells of the immune system, called antigen-presenting cells, seek out foreign cells and engulf them. In addition, despite the best efforts of the immune system, cells can become infected. In both situations, the cell needs a way to communicate with the immune system cells. The phagocyte needs to show what it's captured. The virus-infected cell needs to show that it's infected. Antigen processing and presentation is a way for a cell to display information about its activities.
Okay, so this is MHC class 1. This is MHC class 2. Okay, the purpose of MHC class 1 is to display anything that's inside the body. It doesn't matter if it's virus, it doesn't matter if it's bacteria, like normal cellular function. Anything that's inside the cell will be displayed on MHC class 1, including the virus that infected it. At MHC class 2 are only limited antigen presenting cells, and they only display things they have ingested, or any virus or bacteria they've ingested. Okay.
The process of B-cell activation is called clonal selection. During this response, a B-cell with a specific antigen receptor recognizes an antigen. This cell proliferates to produce a multitude of cells, also called a clone of cells, with the same specificity, which differentiate into memory B cells and plasma cells. This process is called clonal expansion. The antibodies produced by the plasma cells bind to antigens to produce the humoral response.
See what IgM does? Okay, I'll try to explain this using words, and that probably didn't work. But see how like multiple antibodies can attach to these cells, and then like these three bacteria can no longer move away from one another. They're kind of clumped together, and therefore I can come along and kill these three together all at once, and they can't float away and all this other stuff. Okay.
In the simplest form of B-cell activation, an antigen called a T-independent antigen enters the lymphatic tissue and binds to a B-cell receptor, or BCR, on the surface of the B-cell. The B-cell receptors on a particular B-cell are identical and very specific to just one antigen. Once the BCR binds to the T-independent antigen, the B-cell becomes activated. It's now ready to proliferate. However,
see how for T-independent antigens, it has to bind multiple antibody targets at the same time. And the only time where that happens is when you have a lot of repeating units of something over and over again, like polysaccharides.
Most antigens are not potent enough to activate B cells by themselves. In these cases, the B cell will require a second opinion from a helper T cell in order to become activated. We call these antigens T-dependent antigens. The helper T cell recognizes the antigen as it's presented by the B cell and secretes cytokines to tell the B cell to become activated.
Okay. Again, T helper cells recognize MHC class 2. Okay? And they secrete cytokines that activate the cell to proliferate.
Once a B cell becomes activated, it proliferates or divides, forming a clone of cells specific to the same antigen. While some of these B cells differentiate into long-lived memory B cells, most differentiate into plasma cells, which secrete antibodies against the antigen that activated them. The antibodies can enter the bloodstream and travel throughout the body. Plasma cells only live for a few days, but each one can produce about 2,000 antibodies every second, resulting in enormous quantities of circulating antibodies. The antibodies produced by plasma cells have several functions. Since each antibody has at least 2 antigen binding sites, antibodies can bind to more than one microbial cell, resulting in agglutination or clumping. This agglutination helps remove the microorganisms from the body. Antibodies can also neutralize toxins, viruses, and bacteria by binding to critical portions of the toxin or blocking proteins necessary for attachment to cells. Antibodies also act as opsonins for phagocytosis and activate the complement system. Antigen-presenting cells, such as macrophages and dendritic cells, have 2 classes of special proteins that are used to present antigens on their surfaces. Collectively, these proteins are called MHC, which stands for Major Histocompatibility Complex. Class 1 MHC proteins are used to present antigens produced within the cell. A different class of MHC proteins, Class 2 MHC proteins, are used to present antigens engulfed by antigen-presenting cells. Most of the other cells in the body, including infected cells, are also able to present antigens. However, these cells only have the Class 1 MHC proteins. Special antigen-presenting cells, including monocytes, macrophages, and their relatives, engulf invading pathogens and digest them, producing fragments with antigenic determinants. Vesicles containing MHC proteins fuse with vesicles containing digested pathogens. The antigenic determinants from the pathogens are loaded onto the MHC proteins. MHC proteins are used to present peptides on the surface of the cell. Since the peptides are produced from proteins inside the cell, they allow the cells of the immune system to detect proteins inside the cell without entering the cell. Cells in the body routinely break down proteins into small peptides, which are about 8 to 10 amino acids in length. These peptides enter the endoplasmic reticulum and are loaded onto MHC proteins. The vesicles containing the MHC peptide complexes bud off of the endoplasmic reticulum and are released into the cytosol. Vesicles containing the MHC peptide complexes travel through the cytosol to the cytoplasmic membrane, delivering the complexes to the exterior of the cell. If this cell is functioning normally, the immune system cells will simply ignore this presentation. However, if the cell is infected and thus presenting viral proteins, the immune system cells can notice the problem and take action. Each cytotoxic T cell produces a unique T cell receptor, which corresponds to a specific antigen. Each cytotoxic T cell also produces a receptor called CD8, which recognizes the MHC molecules of infected cells. When a cytotoxic T cell encounters an infected cell with a specific antigen bound in an MHC molecule, the cytotoxic T cell produces receptors for interleukin-2 and other cytokines such as gamma interferon, IFN gamma. The cytotoxic T cell can then be activated by cytokines from activated helper T cells. Once a cytotoxic T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these cytotoxic T cells differentiate into long-lived memory T cells, but most mature to attack and destroy infected cells. Cytotoxic T cells can destroy infected cells through multiple pathways. The best understood of these pathways is the Perforin Granzyme cytotoxic pathway. When the cytotoxic T cell encounters an infected cell with the target antigen bound in an MHC molecule, the T cell secretes two types of protein molecules, perforin and granzyme. The perforin forms a pore in the membrane of the target cell, allowing granzyme to enter the cell. The granzyme activates the enzymes inside the cell, leading to programmed cell death or apoptosis. The humoral immune system experiences periods of ramping up and gradual diminishing in response to a foreign invader. After the first exposure to an antigen, there's a lag period in which the exposed person's serum does not contain any detectable antibodies. After several days, the concentration of antibodies slowly rises, followed by a gradual decline. This response is called a primary response. The humoral immune system During the primary response, some activated B cells differentiate into long-lived memory cells. If these cells become stimulated by the original antigen, even years later, they rapidly produce antibody-secreting plasma cells. This faster, stronger response is called a secondary immune response, a memory response, or an anamnestic response.